4. This is chapter 4. The title is The Cell. Can you tell me what you know about a cell already? There's 2 types. Pro and eu. Okay, fantastic. The difference is whether or not they have a nucleus. Fantastic. When I talk about cells, in comparison to what we've covered so far, how does a cell, the topic of cells, how is it different than what we've discussed in lecture so far? It is bigger than a molecule. You're on the right track. It's living. It's the smallest unit of life. It's the smallest unit of life. So this is our first official chapter on life. Okay? Everything that we've covered so far hasn't been living. It's applied to living things. Like we talked about chapter 1, it was kind of an intro. Chapter 2 was elements in the periodic table, chemistry. Chapter 3 was molecules, and then those coming together to make compounds are biomolecules. Okay? Then in the last chapter, chapter 6, I talked about enzymes, which were a type of biomolecule, a type of protein. So now I'm starting with cells, and cells are the basic unit of life. So this is our first chapter on life, technically. Okay? So I have to go over the history of this, but I choose to do it a different way, as opposed to just saying, hey, this is Hook, and this is what he did, and then moving on. I want you to understand it, and I have put a video in here, and I'll tell you how to access it, so when you go to study, you can watch it again. But the only reason we know about cells, and we haven't known about cells forever, is because someone invented a microscope. Okay? The very first microscope that was invented that we get credit for was this microscope right here, Hooke's microscope, a microscope developed by Robert Hooke. And this is in the 1600s, and it is, at that time, the newest piece of technology. So can you tell me what all the townspeople who had financial resources probably did? They went out and bought it. They were like, oh my gosh, that's technology. That's a new toy. I want to go buy it. Just like we do with iPhones and random stuff like that. If it's the newest, we want it, and we'll wait in line for it. That's exactly how this worked. A lot of people didn't even know how to use a microscope, but they still bought it because they had the money to do so, and they were like, oh, I have a microscope. They didn't know what it was for. So we know it, we use it to view very small things, but they were like putting like a shoe underneath it and an apple. Like things that were not microscopic, they were trying to look at with this microscope. And just like a lot of people do who buy all this fancy technology and don't know how to use it, after the new is worn off, what happens to it? Yeah, you put it to the side, it goes to your closet, you find it 10 years later, you're like, oh, I remember when I bought this. And then you try to sell it, but nobody wants it anymore because there's already something new. Okay? Robert Hooke did come up with the very first microscope, and everybody who's anybody bought it. Again, they did start looking at random things, and Robert Hooke himself took a piece of cork, like a bottle cork, that we did in the first lab, and he sliced it really thin, and when he looked at it, this is what he saw. And he just thought to himself, well, I mean, I don't know how he was thinking, but he was like, oh, these look like cells. What type of cells do you think he was referring to? When he looked at that, he said, these look like cells. We didn't know about plant cells yet. Jail? That's what I thought too. I was like, he's talking about jail cells. And then it goes back, and if you watch and read more on the history, he wasn't talking about jail cells. He was talking about monocyte cells. And then I was like, okay, I need more information. But during that time, if you were wealthy, where did you go to school? Like, what did they do for school? Did you go to school right there in your town? What did your parents do? They sent you off to some church school or boarding school where you stayed in dorms, dorm-like structures, but they called them cells. So each student had a cell. And when he looked at these, he was like, oh, these look like the cells. So he called that cells, and now that's where we get the word cell from. Like it had nothing to do with our modern-day animal or plant cells that we're familiar with. It came from him looking at a slice of cork and saying, oh, that looks like a cell. Like, that's it. Like, there was no incredible breakthrough. So Robert Hooke came up with the first microscope, and he called these cells. All right? During this time, Isaac Newton was also coming up with various discoveries, and they were both writing and publishing books. You'll notice as we go through this that there's no picture of Robert Hooke, and it's because in one of Robert Hooke's books, he spoke down to Isaac Newton and actually called him a thief. And so Isaac Newton and his crew stole Robert Hooke's only canvas painting, which is what you did when you, in the old days, if you were rich, you had your portrait, because we didn't have cameras, and they burned it. And so nobody knows what Robert Hooke looks like. So when you do a search for Robert Hooke, a picture will come up and it'll say, hey, this is Robert Hooke, but if you click on it, it's actually a different scientist. Like there's no, we have what we think, we speculate he looks like, but nothing for real, because Isaac Newton and his posse got upset because there's a lot of drama. It's crazy with scientists, okay? Hence, Anton van Leeuwenhoek. Next piece of drama. We do have portraits of him, but just like when we came out with the iPhone and everybody wanted the iPhone, what else came out? The Samsung, right? They were both smartphones. They both technically did the same thing, just different brands. Which one is more popular? Which one was put out first? Which one was introduced to the world first, iPhone or Samsung? iPhone, which is why it's more popular. So Samsung comes along. Anton van Leeuwenhoek comes along and says, well, I can make a microscope too. And it'll do just the same thing. And he did, but his microscope didn't look like that. In fact, his portrait is with his microscope, which is the size of his hand, a really heavy piece of copper. I mean, crazy heavy, and about the size of a looking glass. So like when you had to look at it, you were like, oh, like they were in the old days, and they picked up their looking glass, and they're like, That's what his was like. So he has his fancy little microscope here. You can tell he's quite proud of it. And he, again, was looking at random things like everybody else. But this gentleman scraped his teeth. And I talked about this in our very first lecture. He scraped his teeth, and he looked at it under his microscope, and he said, oh, my, there are animals on my teeth. And he told the world, there are animals on our teeth, and maybe the reason our teeth are falling out is because these animals are eating our teeth. They're living on our teeth, and we wouldn't have never known if it wasn't for my microscope that there are animals on our teeth. What do you think came next once we realized there were animals on your teeth? Then we started doing stuff like a toothbrush. Now, as fancy as we have it now, no. But along those lines, we start to see that kind of stuff happening. So Robert Hooke, he had a microscope. He called cells. We really don't know what he looks like because of the drama. Then we have Leeuwenhoek, who also came up with his own microscope, looked at teeth scrapings and said there are little animals, and he technically called them animalcules, which means small animals, living on our teeth. So that was Leeuwenhoek. 1600s. Fast forward 200 years. We have, what has happened to technology in 200 years? Gotten better or worse? Better. I mean, by that time, Hooke's microscope is probably, oh my gosh, that's one of the first ones. Like, nobody would even admit that they had it. So technology has gotten better and better and better. And we have some German scientists. The first one, Matthias Schleiden, who is a botanist. What does a botanist study? Plants. He looked at plants under the microscope, and he said, all plants are made of cells. I see these cells in plants. So Matthias Schleiden said all plants are made of cells. In the same country, not in the same research institution, but within the same time, we have a German zoologist, Theodor Schwann. What's a zoologist study? Animals. And he looked at animal tissue under the microscope, and he said, all animals are made of cells. And do you recall in chapter 1 when we talked about classification, there was a point in time where if you weren't a plant, you had to be an animal. Like there were only two groups. So at this point in time, plants are made of cells, animals are made of cells. They say all living things are made of cells. Then we have this gentleman, Rudolf Virchow, also German, that initially hypothesized that cells just spontaneously appeared. In our last lecture, what did I tell you does not apply to life? Spontaneity. Life doesn't just show up. So he stared at these cells for a very long time, and he said, oh my gosh, this one cell turned into two. And he watched it happen over and over again. He said, so All cells come from existing cells or other cells. How are you biological evidence of that? Your parents. Your parents are bags of cells. They both took one of their own cells. Those cells came together to create you, who is a bag of cells. I'm here for your self-esteem. Okay? You came from other cells. And we continue to see that over and over again. This is the name of the video. It's on YouTube. It's 6 minutes. There's a lot of sarcasm, so it's right up my alley. But it recaps everything I said, and her voice is kind of funny, and she makes fun of it. Like, it's funny, and it'll help you remember it. But it goes over that whole story of the scientist. She adds a little bit more details because she's a little bit more fun than I am. But I would definitely suggest watching it. It's 6 minutes on YouTube, The Wacky History of the Cell Theory. So here is the cell theory. In biology, we have a few theories that we regularly refer back to, like the theory of evolution, the germ theory. We have the cell theory here, which applies to this chapter. There are 3 aspects to the cell theory. The first, you already know because you already said it. All living things are made of cells. Okay? The second, you also already said the cell is the basic unit of life, or the smallest unit of life. But of course, I put my own little twist on it. I said the cell is the basic structural and functional unit of life. Where do you think I'm going with that? Structural and functional unit of life. Shape is going to tell me what function. Yes. So we have a lot of different types of cells, and they have different shapes. And based on their shape, we can determine their function. Yes. And then the third aspect, all cells come from pre-existing cells. So life comes from life. This is the cell theory that you need to be familiar with. That is glucose. I'm good with that if you want to go to the next one. Okay, before I go any further, I want to know what you think. You already told me there were 2 types of cells. Before we even started, I said, what do you know about cells? Well, there's 2 types. There's prokaryotic and eukaryotic. Let's look at this stuff right here. Pro or eukaryotic? We're going to come back to it, but I just wanted to know, off the top of your head, there was a test question, and I said, hey, I need you to identify prokaryotic versus eukaryotic, which ones you would think. We're going to come back and do this again in just a few. Just your thoughts. These are just different types of cells that have different shapes because they have different... Fantastic. Okay, so we know cells are small, obviously because we have to use a microscope to see the majority of them. Why is it beneficial for a cell to be so small? I'm about to tell you. Cells have to eat and metabolize their nutrients just like you and I do. But the bigger the cell is, guess what they need more of? They need more nutrients. Keeping them small helps them to maintain an effective and efficient metabolic rate. We call it surface area to volume ratio. You don't have to know that. But we keep them small because it's easier, or it's better for us to maintain them that way. How do we study cells? Using microscopes. And you and I have already done that in lab. We've also discussed the different types of microscopes. The compound light microscope is the one that you used in here. We also talked about different types of electron microscopes, the transmission and the scanning electron microscope. In your textbook, it goes over all the different types of microscopes on page 94 if you're interested in going back over that. But we use microscopes to study cells, which is how I started this chapter off. The 2 words I really wanted to bring your attention to were magnification and resolution, which is what a microscope gives us. What is magnification? It makes my specimen larger. So this cell is really small, but when I use a microscope, it magnifies it. So it makes it larger. What's resolution? The clear, the contrast, not necessarily contrast, but how clear, the clarity of it. Yes. So magnification and resolution are characteristics that a microscope gives to us, but they're limited based on the quality of the microscope. The more expensive the microscope is, the better the magnification and resolution. Your microscopes here are around $900 to $1,000, so they're pretty good, okay? But there are some that you can get on Amazon for like $100, and they're not nearly as good as these, okay? But these aren't nearly as good as the best either. Right here I have this diagram from your text that kind of gives you a reference point or a scaling on different things. These aren't all cells, different things so that you can compare. Like, for example, we've studied hydrogen atoms. You can't see them with the light microscope we have in class. You're going to have to use a transmission or electron microscope. Okay? DNA we've discussed is still very small. You can't see it with your light microscope. Cell membrane we've discussed. We're going to do ribosome today. Viruses. But none of these are living. None of these are living. Here, E. coli is living. It's a bacteria. Bacteria. This by itself, these are not living. But you can tell that I can begin to use microscopes, light microscopes, to see them. Here's our typical plant and animal cell. And then a human egg is just a little bit larger. A human egg is a large cell. You never see it, but it has to be large because every female made all of her eggs before she was born. So all of her eggs had to take in a whole lot of cytoplasm, so they are a little bit larger than most of her other cells. Not most, all of her other cells, okay? So when it says typical plant and animal, that's us normally, like our skin and all that. But an egg, a human egg, is much larger because it's been there since you were born, and it has to, in the event that it's chosen, create life for another. You can see frog and fish eggs. You don't really need microscopes to see those. A chicken egg is a cell. A lot of you eat chicken egg, and you have your nucleus. They're the yolk. You have it all there. All your organelles and everything. You just don't see them. You can see the chicken egg. If, and some of you may have had really cool teachers in middle school or high school, they had you take the egg and soak it in vinegar. When you soak an egg in vinegar for 72 hours, it decalcifies that egg. It takes the shell off, and it just leaves the plasma membrane. It's really cool. It doesn't smell good, but it's really awesome. You pick up the egg, and it's bouncy and squishy. Like, that's what the cell membrane is like. It's a really cool experiment if you have a minute. But just so you have an idea. So do all cells have to be super small, like microscopic? The answer is no. And what cell's bigger than a chicken egg? Who has bigger eggs than chickens? Ostriches. Yeah, that's going to be a huge egg, right? A dinosaur egg. Like, those are all really big eggs. So you don't necessarily need microscopes to see all of those. Okay, we're going to start talking about the components of cells. But before we divide up into pro and eukaryotic, I want to address what all cells have. So every cell, so all living things are made of cells. Every cell inside a living organism has these components. So I'm going to give you a moment to get those down, and then we're going to discuss each one. Yes, go ahead.
All cells, all eukaryotic.
Prokaryotic and eukaryotic. Great question. All cells. These characteristics apply to all cells. The first is that all cells include genetic material, or DNA. Some cells have it in their nucleus, some cells don't because they don't have nuclei. But where do you get the DNA from? Where do living cells get their DNA from? Bless you. Say it again, I'm sorry. Where do living organisms get their DNA? Is it just randomly assigned to you? Then where do you get it? Say it again? Okay, that's where it's stored, but where do you get your DNA from? It's a really obvious answer if you're overthinking. Say it again? Your parents. Yeah. I know you're like, what? I knew that. your dad's sperm had half of your genetic material, your mom's egg had the other half of your genetic material. And so you have half your mom's DNA, half your dad's DNA, and when sperm and egg met, now we have you. Now, do you look exactly like half your dad and exactly like half your mom? No. And we'll get into that in a later chapter. We call that dominance and the expression of genes. But ultimately, your DNA came from your parents. Okay? Ultimately, your DNA came from your parents, which is why we call it your hereditary information. So it's passed from parent to offspring. Do you recall when we talk about DNA and we say DNA is your genes, do you remember what a gene is in reference to DNA? It's just a segment. A gene is just a segment of DNA. It's all the DNA together. Genes are just a small piece of that, a segment. Yes, fantastic. Okay? All cells have cytoplasm. Cytoplasm is what we call all of this stuff collectively. Like you're like, oh, that's just in the cytoplasm. Like that's what that is. Cytoplasm. I'm not talking about the nucleus right now because I'm clearly holding a eukaryotic cell, and if you didn't know that, now you do. This is the nucleus, but all this stuff around it is the cytoplasm. The cytoplasm has two components, cytosol and organelles. The cytosol is this clear, semi-solid liquid/fluid. It's like a jello. It's a jello form. And then the organelles are these structures here. So altogether we call it cytoplasm, but cytosol is that jelly-like substance, and the organelles are all of these various structures. When I initially started learning this, I was like, what's the point of the jello? Well, I didn't call it jello at first. I said jelly-like substance. So I started researching, and I found out that this cytosol is put in place so that none of this stuff touches each other. Because it's three-dimensional. It goes all the way around, and if you kind of look here, this one's cut. You can see that if there wasn't something there, they would kind of just stack on top of each other. So the cytosol, the purpose of the cytosol is to hold all the organelles in place. So then I'm trying to learn and trying to remember it. And I honestly thought about my Grammy's pineapple and carrot jello that she makes at Christmas. She makes orange jello, and then she puts a layer of it, and she spreads shredded carrots in it, and puts it in her refrigerator and lets it set. Then she comes back, and she puts pineapples and another layer of jello, puts it in the refrigerator, and lets it set. Because she doesn't want the carrots and the pineapples to mix. So she let the jello serve to hold everything in place. Okay? Now, what do you think happens when that jello gets too hot? It starts to melt, and all those fruits and vegetables mix. What do you think happens when your cells get too hot? Same thing, yeah. So that's why we need to maintain homeostasis. So again, cytoplasm is everything collectively, but cytoplasm has two components, cytosol, which is the jello-type substance, and then the organelles. The last characteristic that we find on every cell is a cell membrane. Do you recall what we also could call a cell membrane? Phospholipid bilayer. What else? This is not good. Cell membrane, phospholipid bilayer, plasma membrane. The last one started with the B. Biological membrane. Okay, and I intentionally, through the notes, change what I call it because I need you to know what. They're all the exact same thing. The different names, but it's all the exact same thing. So all these cells have a phospholipid bilayer. Whether they're prokaryotic or eukaryotic, they have these characteristics. And that was a great question. Pro and eukaryotic, they have those characteristics. So I just wanted to refresh you on cell membrane, also called the phospholipid bilayer. We talked about the characteristics of it, that it has two regions. What did we reference those regions as? This and then that. Hydrophobic. Which part is hydrophobic? The inside. Do you remember what molecules hydrophobic the inside was made of? Lipids. Polar or nonpolar? Nonpolar. So the hydrophobic Lipids are nonpolar. So what does that mean about these phosphates? They're polar, which means they're hydrophilic. When you keep saying this stuff out loud, it's like start making sense in your head. So here's our phospholipid bilayer, which I have titled the cell membrane. Right beneath it, I call it the plasma membrane. And then I call it the lipid bilayer. I am doing nothing but trying to immerse you in a language until you're comfortable. We discussed the difference between hydrophobic and hydrophilic. The whole purpose of a eukaryotic cell membrane is to separate what's outside and inside the cell. But of course, I always take opportunities to pre-teach. And right here, in my phospholipid bilayer, I have proteins that are peppered into it. And we're going to come back to this next week. But in order for this protein to go all the way through, what properties does this protein have to have also? I have my phospholipid bilayer that's both hydrophobic and philic. What does this protein have to be able to do in order to fit all the way through that phospholipid bilayer? What properties does it have to have? It has to have part of it be hydrophobic and part of it has to be hydrophilic. If it wasn't both, it wouldn't be able to go through. If it wasn't both, it would not be able to go through. We're going to talk more on these proteins next week, but I just kind of wanted to throw that in there. Okay, two types of cells. You already knew this. Prokaryotic, eukaryotic. Okay, I put prokaryotes, and then next to it I put my little memory tool, pro-no, and for eukaryotes I put you-do. And even though we say no nucleus and yes has a nucleus, what did I tell you? How did I tell you it was going to be worded academically? We don't say no nucleus. We say, okay, that would be for eukaryotic, but we don't say true nucleus or no nucleus. We say it lacks membrane-bound organelles. It lacks membrane-bound organelles. That's a prokaryote. And a eukaryote has membrane-bound organelles. That's the way we say it academically. As we try to remember it, we're like, no nucleus, yes, it has a nucleus. But technically, it's referred to academically as a cell that lacks membrane-bound organelles and one that has membrane-bound organelles. Okay? On this these are both diagrams straight from your text. You can see that here is a prokaryote. It has the genetic material I told you it would have, because all cells have genetic material. It has a plasma membrane, but it really has nothing else, really structurally. A eukaryote has the nucleus, has all of these fancy organelles. It has structure to it. There are two domains of prokaryotes, bacteria and archaebacteria. There's only one domain of eukaryotes. That's the domain Eukarya. Can you tell me what kingdoms fall into the domain Eukarya? Protist, Animalia, plant, fungi. So basically, anything on Earth that's not a bacteria is a eukaryotic cell. Anything on Earth that is not a bacteria is a eukaryotic cell. So let's do a throwback right quick. Prokaryotic or eukaryotic? Pro. Prokaryotic or eukaryotic? Pro. You. I just want to address the fact that a lot of students say that this is still prokaryotic. Why do they choose that? This word starts with pro. They're like, oh, protist, prokaryote. Don't do it. Protist, you can see that there's some type of shape in there. So that immediately distinguishes it from a bacteria. So a protist is a eukaryotic cell. You were correct with your answer. How do you know it's you? Is it a bacteria? If it was a bacteria, it would be a prokaryote. So algae, what did you say? Algae is not a fungus. If I didn't know, you don't need to tell me what kingdom it is. But if I didn't know, and all I was given was the name algae, and I was told I needed you to tell me if it's pro or eukaryotic. If I know that the only thing on earth that's prokaryotic is bacteria, is algae bacteria? So is this prokaryotic or eukaryotic? Eukaryotic. Okay? And just so you know, yes, algae are protists. So somebody said that, and that is correct. But this is a eukaryotic organism. What about here? Eukaryotic? Fantastic. All right. Everything you need to know about prokaryotes right here. Everything you need to know about prokaryotes right here. Why do you think that I have bacteria next to where it says prokaryote or prokaryotic cells, prokaryote cells? What's the only thing on earth that's prokaryotic? Bacteria. So I'm putting it out there right now. I want you to see that, and I want you to recognize that. All prokaryotes are bacteria. All bacteria are prokaryotic. The very first descriptor is the one that you already knew was coming. Prokaryotes are classified based on the fact that they have no internal membrane structure. They lack membrane-bound organelles. But here in blue are the structures that they do have. Here in blue are the structures that they do have. All bacteria have a cell wall. All bacteria have a cell wall. Many bacteria have a capsule which goes along the outside. The purpose of a capsule, just so you know, because not all bacteria have it, but is that it allows it to hide from the immune system. So if you're infected with a bacteria that has a capsule, it's actually more of a threat to you than a bacteria that is lacking a capsule, because your body can recognize one that is not encapsulated. Having a capsule allows it to hide. So they have no membrane-bound organelles, but they do have a cell wall, and many of them also have a capsule. So those are the first two. You already know they have a plasma membrane, which is also called what? A phospholipid bilayer, a cell membrane, or a biological membrane. Because all cells have that, okay? But I want to make sure you understand what this is saying here. It says the plasma membrane carries out the metabolic functions. Then it says ATP in mitochondria and chloroplast. There is no mitochondria and chloroplast in a prokaryote. What I'm trying to do is tell you that the activities that in our cells a mitochondria would carry out, do prokaryotes have mitochondria? No, they don't. So all of the stuff that our organelles do for us have to be done by their plasma membrane. Their whole metabolism is carried out by their plasma membrane. Bacteria are just one cell. They don't have a bunch of cells like me and you do. So their plasma membrane does all their metabolic functions. The next term up there is the term nucleoid. Because there's no nucleus, we refer to the region where the genetic material is as the nucleoid. The nucleoid is the DNA for that prokaryote. And then right beneath that, it says chromatin and chromosomes. in your body, DNA can be organized in one of two ways. And this applies to all living organisms, not just our body and not just prokaryotes. The first way that it can be organized is what we refer to as chromatin. And this is what chromatin looks like. It's still your DNA, but this is its organizational pattern. If it's not in the form of chromatin, your same DNA will be in the form of a chromosome. So many of you have heard chromosome before. This is supposed to be a circle because this right here is a prokaryotic chromosome. And prokaryotes have just one circular chromosome. Their chromosome is in a circular shape. Does anybody know how many chromosomes you and I have? We have 46 individual chromosomes or 23 pairs, but eukaryotes, like you and I, we don't have circular chromosomes. Eukaryotes, our chromosomes look like this. So we don't say they're circular, we say they are linear. We have linear chromosomes. You and I have 46, but like dogs have 78. I think it's 78. Flies have 12. Like every species has a different number of chromosomes. But if you're a eukaryote, your chromosomes are linear. If you're a prokaryote, your chromosomes are circular. Well, you only have one. I didn't mean to say chromosomes. It's chromosome. Now, let me tell you why it goes back and forth between chromatin and chromosomes. This is the DNA within that prokaryote when that cell is just relaxing and growing. When it's doing what cells do. But do you remember what the purpose of all life is? All life has one purpose, and it's to do what? Reproduce. It needs to survive long enough to reproduce. So this is it when it's hanging out and doing its cell life. Whenever it's time for it to reproduce or for that cell to divide, it organizes itself into a chromosome. The way that I think about it, I reference it, is this is you on a Saturday morning. In your pajamas, your hoodie, you're all just vegged out. Okay, maybe that's just me in a pajamas and hoodie, whatever. But you're comfy. You don't really care. Only close people will see you. You're fine. But when somebody says, hey, Jess, you want to go grab lunch? This is me. I go out in public like I put myself together. Am I the same me? Yeah. It's just this is me like hanging out doing what I do, and this is me going out and being in public. So I'm the same person. It just depends on how I'm dressed and how I'm conducting myself. Like at home, I just study and veg out. And when I'm in public, I have my stuff together, and people are like, oh, she's on it. But it's just a front, right? So here is the chromosome. This is when the cell is dividing, okay, or reproducing. And it stays in the form of chromatin every other time. So when we talk about chromatin to chromosomes, that's what that's referencing. Okay, the next characteristic of prokaryotic cells is that they have ribosomes. This is the only organelle, the only prokaryotic organelle that is also present in eukaryotes. But look at the function of that organelle. What does that organelle do? It makes proteins. Yes, it makes proteins. Protein synthesis. And I told you that, what are we made of? Proteins. All living things have DNA, and that DNA is used to make proteins. So if you have genetic material, which all living things do, you need ribosomes to make proteins. When you were in school, if you ever had to draw one of these cells, when you had to do ribosomes, what did you do? Just like all those little dots, these are all the little ribosomes. They're my little protein factories. Okay, another structure we see in a prokaryote is the flagella. The flagella is used for what, do you think? To move, right, to move. It's used for movement. So overall, I kind of have a summary of characteristics of prokaryotic cells, some takeaways. Relatively simple organism. There's really not much to them. Okay, they're not big, fancy, and complex like you and I. They exploit all known habitats. What does that mean? Exploit all known habitats. I can find bacteria anywhere. I can find them in a volcano. I can find them on your desk. I can find them on your clothes, on your food, on the stairwell. I can find bacteria anywhere. Can I find it in the really, really cold part of the North Pole? Yes, they can live anywhere. Because of that, they vastly outnumber eukaryotes. What does that mean? There's way more bacteria than there are eukaryotic organisms. In one of our first lectures, I said there's more bacteria in this room than there are people on Earth. Like, we're severely outnumbered. And they're incredibly important for cycling biological elements. A lot of people use hand sanitizers and antibacterial stuff, which I get. Because you're like, oh my gosh, I don't want these germs on me. But they are super important. For example, they have a really strong role in breaking down feces. And I bet there's not a human who would offer to do what bacteria can do to feces on their own. Because the way bacteria break down feces is by eating it. I'm not saying that there's nobody who won't do that. But, we're not judging. But I would much rather have some bacteria take care of that job and be grateful that they have that ability. Okay, so they help cycle nitrogen, carbon. They help break that stuff down, put it back into the soil so it can come back and be used by other living organisms. So bacteria are pretty important. Not all bacteria are good, but still. Eukaryotic cells. Eukaryotic cells are in the domain eukarya, and we already know that eu means good or true. Okay, that they do have a nucleus. It includes the protist, fungi, plant, and animal kingdoms. So I have a picture that goes along with each of those. I wanted to point out something that was different. Even though both pro and eukaryotic cells do have a plasma membrane, in a prokaryotic cells, they carry out metabolism. In eukaryotic cells, we don't use our plasma membrane for metabolism. We use it to regulate what goes in and out of our cells. So our plasma membrane right here regulates what goes in and out of our cells. We don't make energy with our plasma membrane. We have mitochondria that do that for us. Okay? Another thing that we use our plasma membranes for is cell-to-cell binding. A bacteria is one cell, just one cell by itself. So it doesn't need to bind to other bacteria. But you and I, we're 100 trillion cells. And if our cells weren't stuck together, what would we look like? Like, I imagine that we would look like a bag of marbles that just spilled on the floor. Like that would be all of our cells just rolling around. Our cells stick together. And a good example or an application of that is if you've ever been sunburned, when you start to peel, sometimes you can peel off like a small sheet of skin. It's because those cells are stuck together. So all of our cells are stuck together, except for our blood cells and stuff like that have to move. But most of our cells, let me rephrase that, most of our cells are stuck together. Okay? Another important takeaway here is that fungi, all plants, and most protists have a cell wall. Animal cells do not have a cell wall. Animal cells do not have a cell wall. So, if I asked you, does a marigold have a cell wall? You would say yes, because it's a plant. Does hummus have cell walls? Yes, because it is fungi. A giraffe? No, because it's an animal. What about a bacteria? We just wrote it on the previous slide. Does a bacteria have a cell wall or not?
Yes.
Yes, it does. Yes, it does. The only cells that don't have cell walls? Animal cells. So you and I, do we have a cell wall or not? No, because we are not to cut you down. But you are. You're just an animal. Okay? So those are good application questions. Don't forget, if it's an animal that can't have a cell wall, pretty much everything else has a cell wall. Some protists don't, but we're not going to discuss those specifically. In your text and in your notes are two diagrams, one of an animal cell, one of a plant cell. Let me tell you what you need to know. You need to know how to label them, both, plant and animal. On your lecture exam 2, you will have a diagram of the animal cell, and you'll have a diagram of the plant cell, and you'll have to label them. You'll be given a word bank, but you're going to have to label them. That's on lecture exam 2. On lab exam 2, not the one you're going to take soon, but on lab exam 2, you'll also have them. And then you're going to have a quiz, the cell quiz. When we do that, you're going to be labeling an animal cell. It'll be 10 questions, and it's just pointing to the different parts, and it says, what is this, what is this, what is this, what is this? So you do need to be familiar with the anatomy of both a plant and animal cell. Okay? There you have our plant cell. Now, we are going to talk about two structures of cells, and then we're going to stop so that I can give you a little bit more time in lab. But I just want to cover nucleus and ribosomes today. All right, so beginning with nucleus. Because we are talking about a eukaryotic cell, they do have membrane-bound organelles. All right? The first structure that is mentioned here is the nuclear envelope. The nuclear envelope here is depicted by this white shell with holes in it. This is my nuclear envelope. And it says that the job of this organelle is to separate the nucleus and the cytoplasm. So this structure here separates the inside of the nucleus and the rest of the cytoplasm. It says it's two membranes. Two membranes. So then I thought, I want them to really get what I mean by this. So here's the nucleus, and I included this whole diagram because of this. And here, this outside part is my nuclear envelope, the purple and green. Then we brought it up closer. Look what it is composed of. Tell me what that is. It's a phospholipid bilayer. It's a membrane, a plasma membrane, a biological membrane right here. So when they say these cells, these eukaryotic cells, have membrane-bound organelles, it truly is that these organelles are surrounded by a phospholipid bilayer, a cell, like a membrane. So I wanted you to see that, and I wanted you to be like, I know what that is. Just because I get excited about random things. So the nuclear envelope separates the inside of the nucleus from the cytoplasm, and it is composed of a phospholipid bilayer. So we can truly say that this is a membrane-bound organelle. It uses the term nucleoplasm. What does nucleoplasm sound a whole lot like? Cytoplasm. There's a reason for that. The nucleoplasm is the cytoplasm found in the nucleus. We just call it nucleoplasm to be more specific. We just call it nucleoplasm to be more specific. So that's the cytoplasm within the nucleus. Of course, you know that the nucleus has our genetic material, and our genetic material can manifest itself as chromatin or chromosomes. And I'm not going to reteach that, but do you understand the difference between those two, chromatin and chromosomes? Yes, ma'am.
Is the chromatin the same in both prokaryotes and...
Fantastic question. The chromatin is the same in both pro and eukaryotes. Chromatin is just referencing the organization, the fact that it's just like everywhere. Okay, and then a chromosome is when it's all tight, nice, and organized. Yes, ma'am. Great question. Then the last aspect of the nucleus is the nucleolus, which is depicted here in this orangish color and a little bit darker purple here. But the nucleolus is responsible for constructing RNA and ribosomes. The nucleolus makes RNA and ribosomes. Nucleolus is RNA and ribosomes. Okay? Questions on nucleus? All right. The ribosome. And if you're using the eCampus notes, this one's not on there. It brings it up in conversation later on, but I wanted there to be a whole slide on ribosomes because we are going to use them quite a bit. So it says here, to begin with, the ribosome consists of a large and a small subunit. So this is the anatomy of a ribosome. Large and small subunit. Then I put their function right beneath it. They are protein factories. What do they carry out? Which is making proteins. Guess where proteins are physically made? The ribosome. The ribosome physically makes these proteins. So we call them protein factories because they are the physical site of protein synthesis. The DNA has the information to make proteins. The ribosomes actually make the proteins. Yes. We can categorize our ribosomes into two groups. Fixed ribosomes, which means that they don't move, and free ribosomes, which means that they do move. The fixed ribosomes are stuck. And we'll learn next week that they're stuck to the endoplasmic reticulum. But they still make proteins. Every bit of protein that the fixed ribosomes make will be sent out of the cell. All the proteins that the fixed ribosomes make will be sent out of the cell. When I think of this and I try to parallel it to something I know, I think about red oak. Red oak has this really big plant, manufacturing plant called Triumph, and they make airplane parts. But do they build airplanes in red oak? No. So they make the parts in red oak, and then what do they do with them? They ship them off to whoever makes airplanes. So the fixed ribosomes, they make proteins that are sent other places. The free ribosomes, they also make proteins. But the proteins that the free ribosomes make stay in the cell. They make proteins for the cell. So I think of these as like your insurance agents. Insurance agents in Red Oak probably have clients that are also in Red Oak. May they have other clients elsewhere? Potentially, but the majority of work that they do is going to be that to serve Red Oak. So fixed ribosomes create proteins that are sent out. Free ribosomes create proteins that actually serve the cell. Questions on the distinction between those two? Okay. Did you have a question? Okay.